Hello, so I'm Anna Krichevsky, coming from Brigham and Women's Hospital and Harvard Medical School in Boston, and I'm also happy to represent the relatively new Harvard's initiative for RNA medicine here. It's really, we are very, very excited about this initiative, which goal is to develop RNA-based therapeutics. I will talk today about glioma. My lab is interested in several neurologic diseases, but this talk will really focus on brain tumors and mostly on glioblastoma, which is the most aggressive brain cancer in adults. I have nothing to disclose. Just wanted to show you a few faces of the key people that have contributed to this work, that what we could capture in one photo. There are a lot more people that I will acknowledge later on in my talk. So we have not talked actually in this session about microRNAs, so I don't want to give you a big introduction, but just to quickly remind you, as the focus of our work is mostly on microRNA, just want to remind you that those are the smallest nucleic acids endogenously expressed, and a couple of key characteristics. Every microRNA can regulate multiple mRNAs, mRNA targets by post-transcriptional repression, both at the level of mRNA stability and translational control. And we are viewing microRNAs today as really master regulators or determinants of cell fate in many conditions, including cancer. Of course, microRNAs are involved in regulation of practically every normal as well as pathological process in practical every cell. So let me tell you a little bit about glioblastoma. This is a terrible disease. It's considered to be an orphan or rare disease. It's extremely aggressive. It's the most common and aggressive primary brain tumor in adults. It affects also pediatric patients. Despite very, very strong research and clinical efforts as well, and numerous clinical trials ongoing at every time, the disease is lethal. So the clinical strategy is very aggressive. It usually includes complete resection or as complete resection as possible, chemo and radiotherapy, but despite this, the median survival is still only slightly more than 1 year. And indeed, the worst part is that it has been very little progress in this parameter, so the survival is changing very little, sort of week by week with different clinical trials. As of today, there are only 4 FDA-approved drugs. The biology of this disease is extremely interesting, but for the purpose of this talk, I really want to mention only one major characteristic, which is a heterogeneity of this tumor. So if you look at the panel of patients or tumors, the histology is fairly similar, but molecularly, there will be huge heterogeneity with many mutations, many fairly uncommon mutations rather than a few abundant mutations. And in terms of signaling, there are at least several major signaling pathways dysregulated in every tumor, and they would vary. I will tell a little more about it later. So overall, GBM is obviously a huge unmet medical need. We have been working on microRNA over many years, and I just want to mention very quickly how we got to the point I will be discussing with you today. So we discovered the first oncogenic microRNA, miR-21, in glioblastoma more than a decade ago, published it in 2005. So miR-21, as of today, is the best investigated oncogenic microRNA, not only in brain tumors, but in a variety of cancers. Fairly early, we realized that microRNAs are involved in practically all aspects of cancer, and glioma is not an exception. So microRNAs regulate cancer cell proliferation, invasion, angiogenesis, and so on. And for the number of years, we are now focusing on one molecule that we believe is especially important in gliomagenesis, which is miR-10B. So today, I will really tell you about this molecule only. This is a slide from many, many years ago, in fact, and published, but even with very early versions of technologies, when we profiled microRNA expression in gliomas versus normal brain tissues, and you can do it by various platforms, of course, you get a fairly consistent results despite the heterogeneity of the tumors. About 30 microRNAs are down-regulated in glioblastoma, about 10 are upregulated, and few of them will be upregulated really across the board in all glioblastomas. MIR10B stands very unique among them. So this slide shows you an analysis of a handful of patients that has been confirmed by the Cancer Genome Atlas that includes more than 500 of glioblastoma and also more than 500 of lower grade gliomas. So as you can see here, and please, I want to draw your attention to the scale, which is logarithmic here. This molecule is really not expressed in the normal brain tissues, just undetectable by tools such as RT-PCR in the brain as well as in the normal cells of the brain, including astrocytes, neurons, neuroprogenitor cells, the cells of glioma origin. It becomes very abundant, one of the most abundant short molecule small RNAs in practically all glioblastoma as well as lower grade glioma starting from the grade 2. So expression of this molecule we consider it as an early event in gliomagenesis. Pretty much the same message, but here I'm showing you what we consider to be glioma initiating cells, or glioma stem cells. The upper panel is normal neuroprogenitor cells, and here is glioma stem cells. They grow in vitro in these 3D cultures, glioma spheres, very, very similarly. They express a lot of the same markers, but MIR10B is a marker of glioma stem cells. It's really undetectable in normal neuroprogenitor cells. Not only this, in fact, when we detect MIR10B in the cerebrospinal fluids of the patients, it tells us with very high confidence that we're dealing with a very aggressive tumor, which can be either glioblastoma or metastatic brain tumors. We never detect MIR10B below a very low threshold level in the non-neoplastic patients or patients with other conditions. So for us, MIR10B is really, as of now, probably the best marker of gliomas. And again, it's not specific to gliomas entirely. I'm not going to talk today about brain metastasis of other cancers. It's a very interesting topic, but it's out of our scope today. So how does it work? And again, this is a very early work. Many years ago, we assessed a panel of microRNA inhibitors or antagomeres, antisense oligonucleotides, and we can use a variety of different types of oligonucleotides. In this case, we used oligos for all microRNAs that are commonly upregulated in gliomas. And MIR10B inhibition was the most significant in inhibiting the growth of glioma cells in vitro, which is shown here. Not only in one cell line, but when you look at the various glioma cell lines as well as patient-derived low passage cultures, MIR10B inhibits their growth uniformly, and it does not affect the growth of normal either neural or non-neural cells that do not express 10B. Okay, so here in one slide, I want to summarize a lot of data that we and other people have developed over the years about the MIR10B function. So as I told you, microRNA obviously regulate multiple mRNA targets. Some of the key targets are listed here, and they include, for example, cell cycle inhibitors such as CDKN1A and 2A. By the way, CDKN2A is one of the key cell cycle inhibitor that is relatively frequently mutated or deleted, rather deleted in glioma. So when it's not deleted, MIR10B can keep the levels of the protein very low. Pro-apoptotic molecules such as BEAM, also MIR10B targets. Interestingly, MIR10B regulates many splicing factors such as, for example, MBNL, MBNL family of splicing factors. So basically when we modulate miR10B expression, we can shift the entire splicing machinery in glial cells. Very important point is that we believe miR10B, despite some very classical targets, the major mechanism of miR10B function is unconventional in a way. And we're only starting at the beginning of understanding this mechanism, but a couple things I want to mention. For example, it's not entirely seed-mediated, so in most cases microRNAs function through the seed of the first nucleotides of 8 nucleotides at the 5' and miR10B seems to work not entirely through the seed or not even mostly through the seed. Also, the majority of miR10B targets are in fact 5' UTR rather than 3' UTR targets, which is also not very common for the microRNA. An additional point here to mention that miR10B function that I will be describing to you today is really specific for glioma cells, despite the fact that it seems to be important for many other extracranial tumors. How it works and what target it regulates is different in these other conditions. So it's a bit of the context effect for this microRNA, which we believe is very important. So it would be impossible to reduce the function of this molecule to a single target or even to a few targets. But when we start dissecting them one by one, some of them certainly are extremely important in gliomogenesis. For example, this molecule, P21, which is one of the direct miR10B targets, and in glioma cells, it binds to the E2F transcription factor, and really by this binding, it modulates the entire transcription machinery. E2F is the major transcription factor that drives the cell through the cell cycle, through the S phase of the cell cycle. So by regulating this molecule, miR10B really regulates a lot of downstream targets involved in the cell cycle. And it's just an example. It's certainly not the single or not even the major mechanism, but it is an important one in glioma context. To put it in a broader perspective, those are 3 major signaling pathways frequently deregulated in gliomas: RTK, RB, and P53 pathways, and those are, as I said, the most commonly dysregulated pathways in gliomas. And as you can see, miR10B regulates numerous key molecules across all 3 signaling pathways. Again, this slide is absolutely not all-inclusive. It just highlights some of the targets that are important for gliomogenesis and their regulation by 10B. Okay, so what happens when we inhibit this molecule indeed? And initially, over the years, we worked with oligonucleotides, and a lot of this work has been done in collaboration with Regulus Therapeutics and their oligonucleotides, which are 2'-methoxy-ethyl antisense oligonucleotides. So to summarize again, a lot of work and the most common phenotypes that we see in practically all cell lines and patient-derived glioma spheres. This is a 3D, my pointer is not really working, but you can see here a glioma sphere, 3D in vitro models of glioma. When we inhibit miR10B by these oligonucleotides, they basically go through various pathways, including propaptotic, and cells just will die and the spheres will fall apart. In vivo, and we can inhibit miR10B by a number of through a number of delivery mechanisms. I will talk about it in a moment, but again, just to summarize, what we normally see is a significant, although transient, inhibition of tumor growth in the orthotopic or intracranial tumors. So, but before we move to this, I want to mention that glioma modeling is extremely challenging. There are no perfect models for gliomas. In mice, we are using a huge spectrum of various models because, in fact, we believe that a lot of problems in the field and the lack of progress in clinical trials is very much largely due to the lack of good models. So just to highlight that we're using established cell lines, we're using patient-derived cell lines are here. They have a number of advantages, including fast growth, but they're really not very representative. They are not similar to human gliomas at all. We're using more and more this low passage patient-derived gliomas that are much more difficult to work with, but they are a lot more representative in modeling glioma. We are also using glioma allografts. There are also transgenic mouse models available, but we have not used them in this project for various reasons. And we have been trying to deliver MIR10B inhibitors locally as well as systemically. So just as an example, and in fact all this work has been published, so I am only briefly going through this. You're very welcome to read if you're interested. So here we deliver this oligonucleotides locally, and as I hope you can see here, MIR10B level is inhibited, and what's more important, and those are again intratumoral injections of the oligonucleotides, we see very nice de-repression of MIR10B targets, and in fact the inverse correlation with MIR10B expression, which results in very significant reduction of the tumor growth and some survival benefit. Again, the effects are not huge. We believe this is mostly due to the very limited delivery, and delivery very much depends on a specific model. So in some models it's much better than in others. Overall, the effects is significant in every model we have tested, but again, it's not long-lasting. What was perhaps even more intriguing or encouraging for us is that our experiments with systemic delivery of MIR10B inhibitors seems to be really promising. So here we used huge doses of the oligonucleotides injected either IV or subcutaneously. And you can see that the oligos, in fact, do go to the brain tumors. So this panel shows you a normal brain with a Cy3 labeled oligonucleotide sort of highlighting the vessels only. Here in this panel, the tumor and the tumor cells show you quite a lot of positive cells. About 70% of the cells are positive here in this model. Not to say that it's common to, again, to all models. So one characteristic feature of glioblastoma is the leaky blood-brain barrier. So it's not a classical neurologic disease that is just impossible to reach by most of oligonucleotides through systemic delivery, systemic injections. In case of GBM, it's slightly more feasible, and again, it very much depends on the model, on the invasiveness of the model, and on the level of penetrance of the blood-brain barrier. What was very interesting that despite the huge doses used here, and we went from 80 milligram per kg up to 150 milligram per kg, so this is absolutely not clinically relevant. Of course, most of the oligos end up in various extracranial tissues such as liver and kidney, spleen, and others. But what we could see again is that MIR10B is inhibited in the brain tumor, and we observe absolutely no toxicity in the, so we observed no toxicity, neither neurologic nor systemic, with these very high doses. Now let me go back. Still, we detect very significant effect. Again, it was transient, but it was significant on the tumor growth, and we have seen a significant repression of MIR10B targets, which is the really our desirable readout. So this was encouraging, the lack of toxicity and the effect. But of course, it brings us to the questions of oligonucleotide delivery. We are in progress, and it will take a huge effort, of course, to develop oligos that are both potent and optimal for brain tumors, for, from perspective of the pharma and clinical toward the clinical trials, but just to remind you, microRNA inhibitors are sort of very similar chemically to the antisense oligonucleotides that are going through clinical trials in various disorders, including the nusinersen that has been approved for SMA, and the number of microRNA modulators are also at the early stages of the clinical trials. Now, while we are thinking about the oligonucleotides as microRNA inhibitors, at the same time we really wanted to ask this key question, whether miR10B is essential for GBM. So again, this is a molecule that is very unique in terms of its expression, expressed in gliomas starting from low grade, absent in the normal neural and glial cells. So we decided to take CRISPR approach to delete it from the glioma cells. And to do this, we developed several sgRNAs, so we could target either a mature molecule here or its precursor. In any case, and we have developed more. I'm showing you a few here. So by doing this, we can hopefully delete this molecule in various cells, and indeed this is fairly efficient. So some sgRNAs obviously are more efficient than others. We clearly seen a correlation of gene editing with the level of miR10B expression or reduction in the cells. Now, when we apply this CRISPR to glioma cells, and here I'm showing you 3 different glioma cell lines in blue, red, and green, a number of controls for some other important microRNA as well as non-targeting, so, and those are miR10B targeting sgRNA, so all of them are able to reduce the growth of glioma cells. And those are single guides, and those are a combination of these guides with Nikkei's version of Cas9. So this version might be potentially a little more specific. Now, let's really look at the cells. So this is our normal culture with one of our cell lines, with all cells healthy, when the Cas9 only is delivered, but no sgRNAs. Here, delivery of 10B, anti-10B sgRNAs leads to this, partial cell death in the culture. So some cells become round, start to float, and then we can analyze them individually. We can look at the healthy cells that are attached, or at the pro-apoptotic cells that are floating in the culture, and what we could see here is gene editing that happens only in the floating cells and not in the attached cells, and correspondingly miR10B levels decreased in floating cells but not in the attached cells. So then we could really do the single cell analysis. So we can take the cells from the culture, raise single cell derived clones, and what we observed was fairly striking. So when we analyzed the single cell derived clones and sequenced the DNA there, what we saw is that all viable glioma cells were wild type. Now when we analyzed the DNA from the cells that became pro-apoptotic, that essentially from the floating cells in our culture, we observed that majority of them, in this case 17 out of 20, were miR10B mutants. So let me reiterate this again. We know that all viable glioma cells express high levels of miR10B. And we know that glioma miR10B mutants are not viable. So this really leads us to conclusion that glioma depends on miR10B for its growth and survival, and provides us the rationale for anti-miR10B drugs. So with this idea, we decided to try CRISPRing miR10B in vivo, and we developed lentiviruses for a proof of principle. And before we go to in vivo, I just want to quickly mention one very interesting thing, I believe, which might be relevant biologically as well as for a preclinical development. So while we were able to edit very efficiently miR10B locus in practically all, in all glioma cells with different efficiency, which is shown here, we practically could not edit this locus in many other cell types that do not express 10B, particularly in primary astrocytes as well as neurons, either human or mouse. So again, this is editing in glioma, and this is with the same viral titers, we absolutely could not see editing in the normal cells that do not express 10B. So probably it tells us a little bit about the structure of the locus and potentially, which is potentially really inaccessible by the CRISPR system in the cells that do not express miR10B. And it also can provide us some sort of therapeutic window, if you will, if you like to think about CRISPRing in clinical purposes, for clinical purposes. Okay, so those are our experiments in one of the mouse models. The tumor, orthotopic GBM is red here, M cherry positive. We can deliver locally CRISPR system. The Cas9 is staining is fairly well localized with the tumors. We're achieving fairly efficient editing. Again, not in all models identical, but in this model it was very efficient. And what was extremely surprising for us after all these years of trying to deliver oligonucleotides, we have seen a huge shrinkage reduction of the orthotopic tumors in these mice. So I'm showing you sort of a fairly typical section, but in some of them we just could not detect the pre-established tumor after CRISPRing miR10B there. So it resulted really in a very strong inhibition of tumor growth and fairly long-lasting, and the rescue of the body weight in these mice. This is another model, pretty much the same result. So about 25% of mice in this case have been just cured, and for others we've seen very significant survival benefit. Now, just to finish, I want to mention another interesting phenomena here. So those are primary mouse or human astrocytes. We can do these experiments with both, that grow in this flask in a monolayer. We can transform them with various oncogenes, such as RAS, for example. So when they transform, they form these colonies. Now, as you can imagine, the normal astrocytes, they don't express 10B. These transformed astrocytes, they start expressing 10B. Now, when we edit miR10B locus at the early stage of transformation, and that's what we've done in this experiment, we can't transform the cells anymore. So in these cultures that have been edited for 10B, we see very few transformed clones, if any, which suggest that miR10B is not only essential for maintenance of glioma cells and for glioma growth, but perhaps it might be really important at the early stages of glioma transformation, or alternatively for viability of this, early transformed cells in the early stage of gliomagenesis. So overall, we think that miR10B is very unique and extremely important in glioma context, and its inhibition by various tools that may include oligonucleotide therapeutics or gene editing may serve as a therapeutic strategy for gliomas. I think the only conclusion that we can really, it would be great if you take it home, that this gene is really unique, and we are thinking about its targeting. We're at the moment, we are optimizing both oligonucleotide and gene editing based strategies for its targeting. We are really trying to do systemic comparison of different modalities. For both strategies, we are very welcoming collaborations. We really hoping to move toward clinical trials fairly quickly, because this disease really needs new drugs tremendously. Let me thank the people that have been involved. There were many collaborators as well as people in the lab. So the three key people that led the work in the lab are highlighted here in red, but also we have multiple collaborators here in different institutions. And as I mentioned, the early work was based mostly on the oligonucleotides from Regulus Therapeutics, as well as our funding sources that are listed here. Thank you very much for your attention.
So, thank you. Interesting presentation. I may have missed that, but you delivered your oligonucleotides either systemically or intratumorally, but did you give it intrathecally?
We have not done it intrathecally, so we have done either intratumoral or osmotic pumps delivery. Again, intratumoral osmotic delivery or systemic. We have not done intrathecal. It's one of the things that we'll probably do with more optimized oligonucleotides.
A couple of questions. The first question is, do you see the difference in on a relapse tissue or a recurrence tissue? Is the levels of the microRNA different? And also the tissues or patient samples who have failed therapy who become resistant, do you see a change in the levels of the microRNA?
Yeah. It's a very good question. So we have tried certainly to find association with miR10B expression with status. So for gliomas, we really don't see a clear association with primary versus recurrent. We also, in terms of the therapies, everybody failed the therapy. So, there are very few exceptions. TCGA has only a handful of long-term survivors. So we clearly see a correlation of survival with the level of miR10 as a family. What I did not mention is that another family member, miR10A, is very, very similar to miR10B. In terms of the sequence, in terms of expression, is different, and miR10B is the dominant in gliomas. So we have seen the correlation of survival with that cumulative level of miR10A family. That's the only thing, only clear association that we found. For the treatments, we could see that, for example, with at the early stage of therapy, miR10B levels in the CSF drop, but when the tumor regrow, we see the elevation of 10B in the CSF. So we believe it really requires more and more longitudinal studies and, of course, a lot more samples. Most of what we have been using is based on a handful of samples available from any single clinic, even such a big clinic as Dana-Farber, and TCGA, which really is basically a collection of everything, and everything fails the therapy at the end.
Does this also exist in the exosomes?
I'm sorry?
Does this also exist in the exosomes, the microRNA?
Whether it is in the exosomes? Oh yeah, so all microRNAs are in exosomes, and this one is not exception. The levels, we can talk about, it's a big topic. We have done a lot of analysis. Yeah, it is in the exosomes.
Okay, I would like to thank the audience, thanks all the speakers for attention. We can close the session. Thank you very much.